Hey everybody, thanks for listening to and viewing our presentation. Our topic is pharmacogenomics. This is Caitlin.
Emily.
And Alan.
To begin with, we would like to start out by telling everybody what pharmacogenomics actually is. Pharmacogenomics is the study of how genes impact the response of a drug in the body. A lot of times this term is used interchangeably with pharmacogenetics. However, there is a difference between the two. Pharmacogenomics is more of a broad term and is the study of how all of a person's genes affect the way they respond to a drug. Pharmacogenetics studies how the body will react to a drug when one of the genes in a body varies. There are a few different types of technology that we looked at in regards to pharmacogenomics. Firstly, a person can get a portion of their DNA tested. In this scenario, microarray technology is normally used. With this technology, DNA is extracted from a person by their blood or urine, for example. The DNA is then separated into two strands that are cut into smaller pieces. The person's DNA is compared to a controlled DNA. These two DNAs are dyed different colors so that they can tell which is which. The DNA is placed on a gene chip, and the two DNAs are allowed to then bind to the synthetic DNA on the chip. Then base pairing takes place, which produces light. Some places on the chip will produce light, which means that genes are present in the sample. It is also possible for a person to get their entire DNA sequence. This allows for scientists to see the order of their nucleotides, or adenine, cytosine, guanine, and thymine. By doing this, scientists can start telling the story of what a person's DNA means. Our entire genome has about 3 billion bases, so one way scientists sequence our genome is through the shotgun method. This method breaks the genome into pieces. The pieces are sequenced and then put back together like a puzzle. Another popular study that scientists conduct is the genome-wide association study. This study allows researchers to see genetic variations that are related to a particular disease. These genetic variations are called single nucleotide polymorphisms, or SNPs. When researchers find these, they are able to assist in providing better treatment to the patient. So now that you guys are familiar with what pharmacogenomics is and some of the technology that goes along with it, we will talk a little bit about the history of it. Pharmacogenomics wasn't really recently popular until the 1990s. People were so focused on pharmacogenetics that researchers really didn't know that pharmacogenomics could be a reality. The major breakthrough for pharmacogenomics was the Human Genome Project. This project allows scientists to map the entire human genome. Then in 2005, the first genome-wide association study was published, which broke open the gates for pharmacogenomics and developed personalized medicine. Personalized medicine is the idea that medicine is going to be customized to each individual's needs by customizing medication types and doses.
Pharmacogenomics laboratories take up a large amount of space that would take away space from existing facilities or require new construction to accommodate a new laboratory on a hospital that would have a lab, which will be very expensive. Recently, Indiana University of Medicine implemented a new pharmacogenomics laboratory, which took up 1,000 square feet of space. Hospitals will also have to decide which equipment they will want to have in a laboratory, because the equipment will also be expensive. Storage of pharmacogenomic information is also a concern. Pharmacogenomic testing will provide a large amount of data. Methods of storing data may include physical paper storage or cloud storage. Physical storage of data kept on paper will take even more room besides just the amount of space for the equipment, which once again will add to being the expensive. Cloud storage will not take up physical space, but it will be expensive to buy the amount of storage needed to store the large amounts of data provided by pharmacogenomic testing. Methods of storage may vary between different facilities, but all information will need to be stored safely and accurately. Before pharmacogenomic laboratories are implemented in a facility, basic education of why pharmacogenomics is important and how it will be implemented must be provided to the clinical teams that will be interacting with the technology and data. The clinical teams that will need to be involved in this discussion include physicians, nurses, and pharmacists. It is also important to educate clinical staff that will be ordering the test and how to interpret the data given by the test. Patients also need to be properly educated about pharmacogenomic testing. Patients who hear about this testing and want to have it done need to be properly educated by their physicians to better understand the process of pharmacogenomic testing and the data it gives and how the data will be used.
Pharmacogenomics has allowed for drug manufacturers to begin researching and developing drugs that are much more effective. The drugs that are being produced through pharmacogenomics are also developed faster, creating an all-around much more cost-effective drug. Due to the fact that manufacturers will have less time and money invested into creating a compound. In addition to this, it will be placed onto the market faster to help people who will need the drug to better their lives. Adverse reactions will occur much less because it accounts for different genes people have. According to the FDA, there are 2 million serious cases reported annually to adverse reactions. Of these 2 million cases, 100,000 people will die. Drugs are supposed to help the patient more than they are supposed to hurt them, and this shows an immediate 100,000 cases where that did not happen. The discovery of this has also led to the total number of patients used in clinical trials to be decreased, due to the fact that they will test the patients beforehand. If the patient will have adverse side effects or the efficacy rate isn't high in them, they will use other patients or fewer of them. Pharmacogenomics also has its downfalls with lost sales and misunderstanding. Pharmaceutical companies worry that they will lose sales on people who do not respond to the drug due to the drugs being tailored to specific genes. Many consumers are also against it because they do not understand the processes and question much about the drug if it is unknown. Pharmacogenomics is a huge deal when discussing the ethics that it holds. Many people are strongly against pharmacogenomics or GMOs that we use daily. Many products, such as corn or other various crops, are genetically modified to help yield a better crop, withstand weather, and immune to various diseases the plants may get. Many people don't like to eat these products because it isn't natural and they believe it is unhealthy for the body, especially in a culture that is becoming more health conscious. Another company dealing with genetics has came under fire recently, and this is 23andMe. The company would actually take DNA from a person and read their genes, and then with this information they would tell the consumer about their health, ancestors, and other various information. It got a lot of attention from telling people about their health and what they are more susceptible to. Many people didn't like this because it was against their beliefs, and they did not like knowing what the future would hold. It could create a panic if somebody is more likely to have a disease that is fatal or very detrimental to a person's health. Many people are also for it because it allows for the chance to take precautionary steps to avoid such diseases if someone is more likely to have them.
Pharmacogenomics raises a lot of questions for the future. Our first question has to do with reimbursement. Will DNA sequencing be reimbursed? If so, by how much? And will all people be eligible to have their genes tested? Another question that pharmacogenomics raises is how will hospitals, clinics, or other facilities where pharmacogenomic testing will take place make money? Initially, the investment in the technology needed for pharmacogenomics is expensive, but the cost being charged to patients is getting lower. How are we going to shrink the payback period for hospitals that purchase genome testing equipment? Will we have to actually raise prices to the patients? Should there be subsidies from the government to pay for the machines? The final question that pharmacogenomics raises is more on the ethical side. Should we require people to have their genes tested? This question is raised because ultimately having people's genes tested will lower the cost of healthcare because treatment will be more efficient.
There are two major business opportunities for pharmacogenomics. Firstly, new entrants into the pharmacogenomic world, which have the potential to grow and generate a lot of sales. If no new entrants join the market, healthcare companies can then consolidate with other existing companies in the same field who are already established in pharmacogenomics. The second opportunity for pharmaceutical manufacturers is to lower their costs of producing drugs. They can lower costs by producing drugs faster, know the demand for specific drug by producing how many people will be infected, and reduce wasted product by knowing when to produce.
After conducting our research on pharmacogenomics, we can predict the future for the industry. Firstly, there will be an explosion of this industry. This is because pharmacogenomics is becoming increasingly popular due to it helping create more personalized care. Researchers are catching the disease someone has before they even develop symptoms for it. Patients' quality of care will increase. Pharmacogenomics will create a lot of ethical issues, but in the end, this type of healthcare is going to save the healthcare industry a lot of money.